Fish parasite is considered a natural occurrence and not a form of contamination. Parasites are divided into two broad categories: roundworm, nematodes, flatworms or flakes, trematode, and tapeworm, castode. Parasite can bring about disease and even the death of the host. White spot visible on skin or fins, high expiration rate, eye bulbs, extreme weight loss, high mortality rate, excess mucus, ulcerative skins. Environment are a huge factor to affect parasite on fish, like temperature of water, stress of fish, pollution of water, polluted feed, high concentration of ammonia in water, and high acidity of farming. So in this lab, we have two objectives. The first one is to identify the types of parasite in aquatic animals. Number two is to observe any clinical signs caused by parasite on the aquatic animal. Assalamualaikum, saya akan terangkan bahan-bahan yang digunakan dalam makmal dalam lab 1. Yang pertama kita ada sushi. Yang kedua kita ada ikan tilapia yang masih hidup. Yang kedua kita ada ikan terisi yang telah mati. Kemudian kita ada tin sardin yang telah ditutupi, so kita tak tahu lah brand apa. And then kita ada dissecting kit, set full. And then kita ada glass slide. And then kita ada cover slide. And then ini adalah distilled water ataupun air biasa. Dan ini adalah selang water. Dan yang ini adalah plate. Petri dish. There's a stranger in bed, there's a party in head. Glitter all over the room, pink flamingos in the pool. I smell like a mini bar, DJ's passed out in the yard. Bobby's on the barbecue, there's a hickey rouge. Pictures of last night, ended up online. I'm screwed, oh well. It's a blacked out blur, but I'm pretty sure it rolled. Damn, last Friday night, yeah we did some tabletops. Now we took too many shots, think we kissed but I forgot. Last Friday night, yeah we maxed our credit cards and got kicked out of the bar. So we hit the boulevard. Last Friday night, we was drinking in the... For the result part, the parasite found in freshwater fish, which is first parasite is nematode, Astrologites, we found at the internal part, and second parasite is monogenia parasite, Dactylogyrus species, which is we found in intestine freshwater fish. The next result is parasite they found in marine fish, which is parasite 3 is nematode, but we cannot identify the species of the parasite, and we found this parasite at the marine fish upper body. Parasite 4, nematode, which is anisakis species, we found in stomach of marine fish body. The last result, which is our group found a parasite 5, which is nematode, and still we cannot to identify that species, and we found in marine fish intestine.
This study obtained two species of parasites found in freshwater fish species of Anabas testudinus, namely Eustrongyllides species of nematode and Dactylogyrus or Ancyrocephalids species of monogenean flatworms. Dactylogyrus and or Ancyrocephalids are known as monogenean parasites that characterized by its specialized attachment organ called hapter and the presence of eye spot. Dactylogyrus are often similar with Ancyrocephalid but can be significantly distinguished by its body shape, attachment structure, and transverse bar. According to Reed, 2009, Dactylogyrus and Ancyrocephalids are the common groups of monogenean freshwater fishes on freshwater fishes. Unlike cestodes or tremathodes, monogenean flugs have direct life cycle by means it transfer from host to host. This species can be transmitted directly from the host. It does not require an intermediate host to reproduce. However, this species is a temperature-dependent in maturation process. The common clinical signs to diagnose monogenean infestations are swollen gills, lethargy, scale loss, extensive skin wounds, and external irritation. Eustrongyllides species are typically known as a big red worm, a fish-borne zoonotic nematode that can be transmitted to the host during the larvae stage. The general characteristics of Eustrongyllides include large, coiled, cylindrical body, pinkish to red in color, and having 12 labial papillae arranged in 2 circles. This species has indirect life cycle that require the intermediate host to survive. The fish that ingested these larvae can potentially cause devastating effects on hosts that can cause gastritis and intestinal perforation, diarrhea, lethargy, etc. Elevated temperature with high organic matter in the water bodies, agricultural runoff, water pollution, overdensity of intermediate hosts in the water bodies are the potential routes contributing to Eustrongyllides infestation.
Compared to freshwater fish, there are two types of nematodes that found in marine fish, which is anisakis species and the other one cannot be identified by species. The detection of anisakis species in the stomach of Nematodes japonicus aligns with previous research showing that this parasite often inhabits the gastrointestinal tract of marine fish. According to Chong et al. 2021, this tract, where the parasite can complete part of its larvae stage before being transmitted to marine mammals. This finding is also important from a public health perspective, as anisakiasis pose a risk to humans who consume raw or undercooked fish containing live larvae. Also, no parasite were detected in canned sardine and sushi samples, likely due to food safety measures such as heat treatment in canned fish and freezing in sushi-grade fish. These processes are known to effectively eliminate parasites like anisakis species, which pose a risk to human health when consuming raw fish.
In conclusion, the study identifies two major parasites, Eustrongylides species and Dactylogyrus species, in the freshwater fish Anabas testudineus. These parasites, which have different life cycle and environmental requirements, pose ecological and health concerns to fish population. Dactylogyrus species, a monogenean with a direct life cycle, thrive in temperature-sensitive environment, cause gill injury and skin irritation in fish. While a Eustrongylides species, a zoonotic nematode, requires intermediate host and is associated with water contamination and organic materials, inflicting significant internal harm to its host. The finding of anisakis species in marine fish raises public health concerns, particularly for people who consume raw fish. However, food safety techniques such as heat treatment and freezing serve to limit these hazards. Therefore, effective environmental management and several food safety procedures are required to reduce parasite illness in fish and avoid zoonotic transmission to humans.